So the lab has basically two main, I would say, foci, two main areas. One is mammalian cell structure and function, which is basically focused on cell migration, on sort of tumor cell growth, which is one of the smaller parts now. It used to be a bigger part, but for a variety of reasons, which I'll talk about. It's a smaller part, and the second one is global health, more recently focused on development of technologies, but also basic science questions affecting not just global health, but more recently refugee health as well.
Yes.
So if you look in sort of almost the division of the lab, on the right-hand side there we have sort of the mammalian cell culture side, which is basically you'll have sort of just regular cell culture, and I'll talk about the microscope that we have, and we've done a lot of sort of work in understanding how cells migrate in three-dimensional environments. So native like tumor environments in terms of their mechanics, interactions, cell structure, cell function. So the other part of the lab, which is sort of separated a little bit to make sure that there's no contamination because we work with bacterial systems there, is focused largely on global health questions. So one of the things that we are very interested in is this question of poor quality or substandard medicine. So one of the things that you would see in low and middle income countries is that the qualities of medicine, for example, antibiotics, antimalarials, may be compromised. And that impacts those communities in sort of affecting the quality of their care. So it develops drug resistance, of course, mortality and morbidity, financial loss. So we are very interested in understanding the biology of that question. So what happens at the bacterial level? What happens at the pathogen level in terms of the resistance patterns that may emerge, but also interested in solution space. So one of the technologies that we've been working on for quite some time is being able to test the quality of medicines, excuse me, across the board, and do that at the point of care. So a technology, PharmaChk, and you see sort of the guts of that device here, which is sort of an optical / microfluidic system where somebody can just put in the drug and the system sort of takes care of it. We have the aptamers and the molecules that would bind, give a fluorescent signal, and tell you quantitatively whether a drug is has the right quality parameters or not. And you see sort of the inside guts of the device. You can see that it's the size of a laptop, a suitcase, not particularly heavy, that can be taken to all points in the supply chain.
Yeah.
So those are the kinds of questions we are interested in, largely driven by this idea of thinking of engineering and engineering approaches broadly and holistically in addressing important questions for those who are historically marginalized, people in low and middle income countries, refugees, people who are sort of forced out of their homes because of conflict or climate change. So we start with sort of the larger set of things. We want to make sure that all of our questions are first scientifically robust, which means that we first make sure that the reactions are taking place and sort of think of it at macrofluidic or sort of at the test tube level, and then miniaturize it, right? So there's the science part of it, the discovery part of it, and then the engineering and miniaturization part of it, and we've done both of those. But I would say that when I say we, it's not just my lab. We've had partners all across the world in industry, in sort of NGOs, and on the ground who've sort of helped us do that. So let's take a look at how the thing works. So right now you're seeing sort of the back end of the device. So this is sort of where the guts of the device are, various controllers that allow sort of the movement of the fluid, the sort of the light control, sort of making sure that the device is able to function and give off signal compared to the controls. And let's say you are interested in looking at antimalarials or anti-TB infectives and are able to sort of procure them from the market, and you want to see what is the quality of those.
Yeah.
So you would put the drug, and then it would bind to a specific molecule that we have worked on. So those could be small DNA snippets, those could be small molecules. Upon the reaction, they'll give off a signal, and the signal is proportional to the concentration of the active ingredient. So if the signal calibrates to 100%, then the drug has 100% of the active ingredient. If the signal responds to 35%, so it gives you a quantitative information in a matter of minutes. So you don't need to use an HPLC, you don't need to use something that is both expensive and requires large infrastructure. The whole thing can just fit into this system as well. So on that end is you see the inside of what this is. So the whole thing there fits into this, and you can sort of just take it with you at the point of care.
How heavy is this?
It's about, I would say, between 15 to 20 pounds. So it is within the airline allowance for overhead.
Yeah, I actually watched one of the video in one of the lectures you had. You say, I think it's like WHO or one of the health organization is like way over 100
pounds. That's right.
Yeah.
We have it somewhere here. I will find it. We have it here somewhere. And I don't know if we have it or if we've returned it. But the challenge was that to be able to take it not just at the point of care. No, I don't think we have it here. Not just at the point of care, but to be able to take it between points of care, from one hospital to the other, from one sort of environment to the other. So let's say you want to test something on the port of entry of a drug at the port, and you want to do the same thing at the hospital, in the dispensary, in the pharmacy. So you want something that can be easily collapsed together and taken on a motorbike or in a car or however you see it. So that was important. The other thing that I think is important is to be able to get the answers in a short period of time with minimal infrastructural needs, and something that is fully self-contained.
That's awesome. That's a drastic improvement.
Well, we hope that it can sort of really start to be implemented, but that's the hope.
Yeah, that's awesome. Okay. Do you want to show us other part of your lab?
Yeah, so another project that we are very interested in is antimicrobial resistance, particularly in the context of low and middle income countries. And on this side, we sort of just do that. You'll see sort of typical shakers where we are culturing these and trying to understand what exactly is the role of the environment. Again, another thing that we know, and I think it's becoming more and more important, is the fact that climate change impacts the transmission of disease, and that's not what we're looking at. But climate change also impacts the lives of people living in urban slums or in low-income communities or in refugee camps. And there, the presence of many of these factors influences the development of drug resistance. Another area that we are seeing now in Ukraine, but also elsewhere, is the presence of conflict. So conflict itself drives infection because people get injured and die, but also it creates an environment where you have heavy metals that go into the environment, and those heavy metals may also impact the development of particular kinds of resistance profiles. So there are some important unanswered and I think globally relevant questions in drug resistance that we are very interested in.